Hello class and welcome to Lecture 16. Today we will begin Unit 3 or Module 3 of our course. I'll spend a little time introducing that when we get to the next slide. The topic of the day is an introduction to signal transduction. And what signal transduction is it's a way of cells communicating with each other. It involves extracellular factors binding to surface receptors and the induction of a signaling cascade inside the receiving cell. We call this signal transduction, and we're going to focus on this quite a bit in our third unit. In order to sort of set the stage for the third unit, we're going to first introduce the concept of signal transduction, and then I'll give you some insight into the key players that get things started, the ligands, those are the extracellular factors, and then the receptors at the surface of the receiving cell that set off the transduction cascade. The reading is from Albert's chapter 16. So, if you think back to our last lecture, that would be lecture 15, that was sort of a standalone lecture. In that lecture, we covered a paper related to the cloning of the beta secretase enzyme. And it allowed me to sort of dive deep with you into that topic. So now we're done with Unit 2. We've had our transition sort of standalone lecture. And now we begin Unit 3. So the first thing I'll do by way of outline here is I'll first introduce Unit 3 to you, give you a sense of the overriding theme, and then I'll go into an introduction to signal transduction, talk to you about how cells signal to each other. And I'm going to give you a historical perspective, as I often like to do, and talk to you about the early discoveries with epinephrine, a signaling hormone, and what it does to receiving cells to cause the breakdown of glycogen and make available glucose. We'll then go through the major varieties of cell-cell signaling, and then go through a laundry list of ligands, and that will bring us to the end of part 1. So this is part 1 of my lecture today. In part 2, we will turn our attention to the types of receptors. We're covering the major types. And then I want to spend a good bit of time at the end of the lecture, part 2, talking about a particular technique for identifying receptors for so-called orphan ligands. And what we'll see is it's a return to the technique we talked about in our previous lecture, the expression cloning strategy, where you use a cDNA library, express it and turn it into protein, and go fishing for a particular receptor for a ligand that you might be interested in. So we'll begin with context, we'll end with experimental approach. Okay, first, context. Remember that it's always important for us to be thinking about the story or the narrative of the course. I've asked you to think about this course as a book with 3 parts, and each part of the book is a module of the course. And each lecture really is a chapter within that part of the book. So let's think about where we are and sort of what we've covered to bring us to this stage. We began Module 1. Module 1 was defining the cell and its study. And here we were essentially looking at the prototypical fundamental unit of life, the cell, and focused on its structure function. So if you think about some of the topics that we talked about here in Unit 1, we talked about the way that the cell is structured. We talked about how the membrane is structured, the proteins in the membrane, moving things across the membrane. We looked at how material moves towards the surface and gets endocytosed back in. We looked at the internal structure of the cell, the cytoskeleton. We looked at how we fuel the cell. We also talked a lot about experimental approaches, model systems, how we clone genes, things like that. What that was doing was it was setting the stage for everything that follows. That was sort of our introduction, if you will, to the course. Now, Unit 2 focused on information flow. So it was really organized around the central dogma. And it really covered the mechanisms of DNA transcribed into RNA translated into protein. And of course, we looked at replication of DNA as well. And it really focused on how all of these steps are regulated. We also, of course, talked a lot about experimental approaches in this unit. And we ended that unit with a focus on proteins and how they are modified post-translationally. So things like proteolysis, phosphorylation, etc. Now that we're up to the protein level, Unit 3 is going to look at protein pathways. And really what we're thinking about is a network in the cell of various proteins that function together in chemical pathways. So you might have some upstream activators of protein X. And protein X then activates protein Y, and protein Y activates protein Z, and protein Z causes some effect. This protein pathway or this protein network is what we are referring to when we talk about signal transduction. Particularly when this intracellular protein network that I've indicated here is set off by an extracellular ligand. So a ligand from outside the cell, released from a different cell, perhaps even somewhere else in the body, is acting on receptors at the surface of the cell that then initiate the activation of this pathway. So that's what we're all about for Unit 3. We're going to, of course, cover a lot of the detail of the signaling pathways. As you know, this course is very detailed. We're also going to be focusing on a lot of the experimental approaches that help us to learn about these pathways. By the end of it, you'll be very well versed in designing experiments and thinking through the logic of the protein pathways. At least that's the goal. So that's the introduction to Module 3, Protein Pathways. Now let's begin the introduction to signal transduction. So here's the overview, and I just want to say before we get started, this is one of my favorite topics in all of cell and molecular biology because of its elegance and really its widespread importance. You'll get a sense of that as we move through the material. It's really pretty cool. So signal transduction is all about this interaction at the surface of a cell where an extracellular signaling ligand, you may have heard other people pronounce this as ligand, potato, potato. I pronounce it ligand, where a ligand from the outside binds to a surface receptor. And you can see that surface receptor, they've shown it in green here, and I've just drawn all over it, but you get the idea. It's membrane anchored, it's localized to the plasma membrane, it's going to be anchored to actin cytoskeleton most likely, and it's got an extracellular domain sticking out here in the extracellular space, waiting for the ligand to bind. And when that ligand binds, it changes the shape or the conformation of this receptor. And that is what sets off the primary induction or the primary transduction event. So ligand binding to receptor, changing conformation is primary transduction. And what that does is it now taps into this protein network, which is shown here by all these red arrows. Each of these red arrows represents an individual protein in the network. So these proteins would be the upstream proteins. We think about the flow in this direction. And any of these processes or proteins would be downstream. So upstream, closer to the receptor, downstream, further away. And along the way, we have typically a relay, and that was the XYZ thing that I was indicating on the previous cartoon. Other things that can happen along the way in the network is amplification of the signal, where we go from a sort of one-to-one relationship to maybe a 1 to 100 and then a 100 to 10,000, some kind of dramatic amplification, chemical amplification in the pathway. And then another feature is often divergence. So often upstream mediators like this can then kick off 3 independent downstream pathways. And that allows there to be a lot of diversity with regard to the effect on the target cell. So the same ligand can end up having 1, 2, 3 different effects in the responding cell. It's pretty cool. A lot of versatility. So another way of thinking about signal transduction is to say things like ligand receptor interactions produce an intracellular change. And that intracellular change can be short-term, long-term, or sometimes even permanent. So transient or short-term events happen in milliseconds to maybe tens of seconds timescales. Long-term changes would be, in the range of tens of seconds to minutes. And then permanent changes obviously are long-lived through the lifespan of the organism. Think things like puberty, for example, and the hormones that allow for those changes. Other things to think about, a lot of times the target of a signaling pathway is a change in gene expression and a change in the way proteins are produced and then sometimes turned over. So these signaling networks can feed back onto information flow. They can hit it at the DNA level to change transcription of genes. They can hit it at the mRNA level to change the turnover of a transcript. They could even affect it at the protein level by affecting its translation or its modification after translation. Okay. One last sort of thing I'll leave you with, and this is somewhat philosophical, I guess, but if you are a cell, you're only as good as the complement of receptors that you express. And what I mean by that is there's no way for you to respond to an extracellular signaling ligand if you don't express the receptor for that ligand. And an analogy that I often will use to illustrate this idea is, imagine that you get into a car and you're turning on the car and the stereo is playing, and you decide that you want to change the channel. So you tune the radio to a different frequency to pick up a different signal. Now you're listening to 104.1 or whatever instead of 97.9. So the tuning of the radio is sort of analogous to the receptors that you're expressing, and they're allowing you to pick up the signal, the frequency of the signal if it's a radio signal, and that frequency is analogous to the ligand. So the only way to pick up, if you're a cell, a different type of ligand is to express that receptor. So the complement of receptors is absolutely key. So when we cover signal transduction in this course and in most courses, we tend to focus on metazoans, animals, and that's because animals really do this in a major way. But we should not ignore the fact that cells from all organisms do signaling. And I'm going to give you sort of a simplified phylogenetic approach to this here, just to give you some simple examples in other organisms, so that we don't think of this from a metazoan-centric way. For example, single-celled organisms will do biosignaling, but typically what they're doing is sensing the environment. So they'll have surface expression of receptor-like molecules, bacteria will do this, that will allow them to sample the pH of the environment or look for oxygen in the environment, for example. So it's very simplified. There doesn't tend to be as elaborate intracellular networks in place in the prokaryotes, but they do simple signaling. Plants, multicellular, non-animal, obviously, but very, very robust signaling pathways exist in plants. One that we're all familiar with, especially now that spring is, I guess, finally coming to Kalamazoo, is the idea of phototropism. So, you may notice that you've got plants that are starting to bend towards a source of light. They will always do this, that's phototropism. And the chemical mediator is something called auxin. And auxin's a hormone that allows for shape changes in these columnar cells at the tip of the plant. And when those cells change shape, it allows bending, and they will always bend towards the source of light. So that's an example of a signaling pathway. In a way, light is the ligand that sets off the signaling pathway that involves this hormone auxin. Animal cells really do this in a big way, and that will be our major focus for Unit 3. And we're going to see a wide range of signaling factors that can induce signal transduction cascades in animal cells. Another sort of interesting feature, or not feature, but fact, I guess, is signaling is very important clinically. And we've learned over the years that quite a few transforming events that can turn a normal cell into a cancerous cell involve changes or breakdowns in signal transduction pathways. We're going to cover a little bit later a protein called RAS, and RAS is a very important protein when it's mutated in colon cancer. And we're going to see what changes to RAS lead to some types of colon cancer. So that is by far one of the more intriguing aspects, at least to me, of signal transduction biology, is it gives us some insight into some clinical issues like cancer. So more about this later, but that's a little preview that I'll give you and a little tease. Okay, so we've done the overview. We've told you, or I've told you, that all cells really do it. Now let's take a look at one of the earliest signaling pathways that was discovered. And the early work on this dates back to the 1950s. And some of these names will be familiar. Krebs, this is the same Krebs who discovered and described the Krebs cycle in oxidative phosphorylation. So one of the things that these scientists were interested in was how extracellular epinephrine can lead to changes inside a cell. Much of their work was done in cultured cells. They were culturing either liver or muscle cells. And they found that if you take a culture of cells and you add to the culture medium a hormone called epinephrine, so you add epinephrine to the culture media, you can detect reliably changes inside the cells. And one of the changes that they noticed inside the cell was a rise in a particular type of cyclic nucleotide called cyclic AMP. This is what you get when a cyclase converts ATP into this cyclic AMP cyclic nucleotide. So they had no idea how epinephrine from the outside of the cell led to an intracellular rise in cyclic AMP, but they were able to describe that correlation. They started then looking at what other changes they could find inside these receiving cells. And one of the things that they noticed was a breakdown of stored glycogen and an increase in the amount of glucose available for glycolysis. And so they reasoned that epinephrine acting from outside the cell must be somehow engaging a molecule at the surface of the cell that then led to this rise in cyclic AMP, and then eventually, through an unknown mechanism, allowed for the conversion of glycogen to glucose. Now, it took about 25 or 30 years to work out all of the details, but eventually this cascade that this cartoon is indicating was discovered. And let me just kind of walk you through the elements of the cartoon, give you a sense of what we're talking about here. So this would be the the extracellular space, and this would be the cytosol. So we're looking at the plasma membrane here. You see the lipid bilayer. Here is the receptor for the epinephrine hormone, and it has an extracellular portion that has a ligand binding domain. A ligand binding domain is the part of the epinephrine receptor that engages the epinephrine, much like a lock and key type of mechanism. Well, that binding of epinephrine to its receptor will change the shape of the receptor. And it will start inducing, or I guess what I would like to say is it'll start allowing that activated receptor to tap into an existing protein network. And that existing protein network involves things like adenylate cyclase. That's one of the first enzymes. It's not shown in the cartoon, and it's not this purple thing. It's represented by this arrow. Okay, and later on I'll actually show you a schematic with that cartoon drawn. For now we're just thinking about it schematically. And what the adenylate cyclase does is it converts ATP into cyclic AMP. And then cyclic AMP levels go up. And then they can activate a protein kinase called PKA. So now this is active PKA. And notice how the artist has drawn this little halo of activity around it in sort of yellow. That's to remind us that when cyclic AMP is around, this enzyme is now active. Okay? So now protein kinase A is going to start to work on its substrates now that it's on. And like all kinases, PKA is going to transfer a phosphate from ATP to its inactive substrate. Okay? And what that results in is the active version of that substrate. And this substrate's name is phosphorylase kinase. Since it's also a kinase, it's going to go and transfer phosphate from ATP to its inactive substrate, turning on that downstream target. This one's called glycogen phosphorylase. And then glycogen phosphorylase causes the conversion of glycogen to glucose 1-phosphate. Okay? So this is a signaling pathway, and don't worry at this point about really committing to memory the nitty-gritty details of this. This is simply serving as an illustration of what we're building up to. So we're going to cover this exact pathway again, don't worry. I just want to give you some insight into what we would be looking forward to in the third unit. And this is a good place to start because historically it's one of the first signaling pathways we ever found out about. Okay, now that we've looked at sort of the introductory segments of the talk, now let's take a look at how cells do communication and the varieties of intercellular signaling. In other words, how does this ligand-receptor interaction work? And how many different varieties has evolution come up with to solve this issue of cells talking amongst each other? And we're going to begin sort of over here with direct cell-cell signaling. And this is one that I think is really kind of cool because here the ligand and the receptor are both membrane anchored. So one cell is expressing the ligand, the little green guy that's at the surface there you can see. And the second cell is expressing the receptor, the little red protein at the cell surface. And when these two cells are in proximity to each other, the ligand and receptor can engage, and that then allows signaling to happen in sometimes both cells, but certainly in that second cell that is expressing that receptor. So in direct signaling, the ligand is never released from the ligand-producing cell. It's not secreted, it's just simply anchored at the cell surface, and it interacts, in that context with its receptor on another cell. We often see direct cell-cell signaling in the immune system, particularly with innate immunity, and we also see it quite a bit in development, early in embryology or embryonic development, when there isn't a lot of complexity in the tissue. So before things get really complex and we're just dealing with a few cells, direct signaling is the way that the transduction events typically happen, and the intercellular signaling typically occurs. Now, once we're out of the developmental window and we're into more complex, mature organisms, these other three mechanisms come into play. And we're going to begin with this one in terms of those other three. It's called paracrine signaling. Paracrine signaling happens when a ligand is secreted from a ligand-producing cell, and then it travels over a short distance in the same tissue, typically, to then bind to a receptor on a neighboring cell. So this would be the ligand-producing cell. The little blue sphere is the ligand. It comes through the secretory pathway. It gets released. Now it's in the extracellular space. and it travels over a short distance, kind of following typically a diffusion gradient, until it finds a receptor to bind to on a neighboring cell, and then induces that primary transduction event, and then allows the receiving cell to have some effect, causes some effect, I should say. We see this a lot in the nervous system. So chemical neurotransmission across a synapse is a good example of paracrine signaling, signaling over short distances. Okay. Some hormones work this way too within tissue. Okay. Now let's move over here. So sometimes a ligand is secreted from a ligand-producing cell, but instead of traveling over a short distance in the same tissue, the ligand instead makes it into the bloodstream, and then it travels through the circulatory system to a distant target. And the ligand will then have to move out of the bloodstream back into the extracellular space, often in another organ of the body, and then bind to a receptor on a target cell and have an effect. So we call this endocrine signaling. And endocrine signaling involves the bloodstream, and it happens over much longer distance. So the classic sort of key hormones that we think of, testosterone, estrogen, progesterone, these all are endocrine factors that travel in the bloodstream and often have an effect at a distance. Other examples would include anything secreted, say, by the pituitary. And often the pituitary gland secretes hormones that have an effect on the adrenal gland. And you probably know the adrenal's on top of the kidney. So you can't get much further away, or it's a pretty good distance at least, between the base of the skull where the pituitary is and the renal system, the adrenal system. So endocrine signaling, like paracrine signaling, but instead of short distances, larger distances. And then the last one here, this is kind of the weird one, autocrine signaling. And in this one, the same cell that produces the ligand and secretes it also expresses a receptor for that ligand, and it will bind to that ligand, and that will induce a signaling cascade in the receiving cell. We see this a lot in the immune system, and particularly with adaptive immunity, and lymphocytes are a good example of cells that do this a lot. And you might wonder, why can't the ligand just have its effect inside the cell? Well, the ligand needs to bind to the receptor at the surface, and that has to do with the proteins around the surface of the cell that the receptor is hooked up to. So it's not possible for the ligand to interact with the receptor inside the cell and have the same effect. It needs to happen in the context of the anchored receptor protein. So autocrine signaling is another variety. So these four varieties of intracellular signaling, these are ways that cells have evolved a way to communicate with each other. Ligands released from one cell, binding to receptors on another cell. All right, so now let's turn our attention to the types of ligands. I'm going to cover a few with you. We're going to begin with the steroid hormones. So steroid hormones are somewhat unique among the different types of ligands. They typically are large and hydrophobic, and so they don't often need, in fact, they don't typically need, a cell surface receptor to work on a receiving cell. Instead, the steroid hormone, and here's the outside, here's the cytosol, here's the membrane, can simply slip across the membrane to gain access to the receiving cell. And heck, it can even slip across the nuclear envelope, as you see it doing here, coming into the nucleus. And there, in the nucleus, it will bind to its receptor. And for this particular steroid hormone, it looks like you need the ligand, the green dot, binding to the receptor, and then two of those come together. So that would be homodimerization, two of the same subunits bind together. And you can see what they're doing. They're acting as a trans factor to sit down on a cis element to drive transcription. So what we'd be dealing with here is a promoter. So the cis element would be a DNA sequence that we think of as a regulatory sequence think promoter. And the trans factor is the protein that binds to the cis element, and in this case regulates transcription. Anytime we see this kind of an arrow, we're indicating that transcription has happened. Now look, a lot of stuff's not shown here, right? We don't see the RNA polymerase, the whole complex coming, the whole transcription factor apparatus that allows the unwinding and the transcription, but it's implied by that arrow. So steroid hormones are very powerful, and one of the reasons is because they directly impact gene expression or transcription. And these are examples of ligands and types of signal transduction pathways that can have lifelong effects, particularly when talking about the hormones of puberty. Here's another example. Here, the trans factor's already homodimerized and already exposing DNA binding domains that allow it to interact with the promoter, but it's acting as a transcriptional repressor because it's not in the right shape yet to recruit the RNA polymerase. So what puts it in the right shape is the steroid hormone. So now when that comes into the nucleus, it binds to the trans factor, changes the shape, and look at what the artist has done. Here, this little activation domain is hanging out like that. Now it's flipped down, okay? That's the conformational change. Now that trans factor is in the right shape to recruit RNA polymerase and activate transcription. So again, no matter what the sort of specific mechanism is, a hallmark feature of steroid hormones is activation of gene expression. And typically, it just puts the trans factor in the right shape to allow it to either bind, as in this example, or recruit RNA polymerase, as in this example. Either way, we're impacting gene expression. This may surprise you. I remember learning about this in grad school and being kind of surprised by this. But gases can also play a role in signaling. Not every gas by any stretch, but certain ones, like carbon monoxide, believe it or not, or nitrogen oxide. Both of these are known signaling ligands. And they typically work in a paracrine manner. So let's take a look at one example of a gas working as a ligand, and this is the dilation of blood vessels. So anytime we want to increase blood flow to an area of the body or to a particular tissue or organ, we dilate the blood vessels that feed that tissue or that organ. And the way this happens is by the relaxation of the smooth muscle cells that surround the cells that make up the blood vessel wall. So here's a look at it. We're looking at a blood vessel in cross-section. The blood would be traveling here in the lumen. And these yellow cells, they're called endothelial cells. They're sort of fused together and they make up the lumen of the vessel. Surrounding those endothelial cells are smooth muscle cells. They're the ones shown in gray here. And there's several layers of those smooth muscle cells. So if these cells, these smooth muscle cells, constrict or contract, they tighten up the blood vessel lumen, and less blood can flow and it flows under higher pressure. If those smooth muscle cells, instead of contracting, they relax, they spread out more, and that allows the lumen to expand. Because whatever the smooth muscle cells do, so will the endothelial cells. So here's the mechanism. It's really kind of neat. There is nervous system innervation of the vasculature. And in blue, we see a nerve, an axon you can think of, coming in and starting to connect with the system. Now, the artist hasn't done a great job here. It's good, but not great. I'd like to see these blue fibers threading in and synapsing on those yellow cells instead of making it look like the synapse on the gray cells, but we can work with it. Over here is a little more detailed description of what I'm trying to describe here. So this would represent the nerve, and at the end of the nerve is the axon. And here is the synapse connecting the axon to the endothelial cell, which is the yellow one in this diagram. So you can see that a ligand, they're showing it in yellow here, is released from the nerve, or the axon. It binds to a receptor on the endothelial cell. And I've kind of obscured it, but you can see two little arrows here. it sets off a signaling cascade. And the result of the signaling cascade is the conversion of arginine into nitric oxide. Nitric oxide, NO, is the gas ligand. And it's going to diffuse rapidly across the membrane. It's small enough it doesn't need anything to get it out. It just diffuses right across the membrane and into the cytosol of the smooth muscle cell. There it binds a target protein. Think of that target protein, they're showing it in blue here, as sort of analogous to a receptor for nitric oxide. And I'm not going to cover this mechanism with you, but suffice it to say that this induces another cascade in the smooth muscle cell that eventually causes the relaxation of that muscle cell. And now it spreads out, and that allows the lumen to expand. Okay, so it's pretty cool. The mechanism isn't so important. What I really just want you to get is the idea that a gas can act as a ligand, and when it taps into an appropriate signaling transduction network, we can get pretty dramatic effects, like a total shape change for a cell that this protein works in. So the gas works in the smooth muscle cell, causes it to change its shape and to spread out and relax and have pretty dramatic effect. Okay, so here's another paracrine type ligand, and this is the neurotransmitter. And obviously the neurotransmitter is the key mediator of communication between neurons, but not just that, sometimes between neurons and other target cells like muscle cells. In fact, you just saw an example in the last slide of where a neurotransmitter, acetylcholine, is having an effect on endothelial cells, the kinds of cells that line blood vessels. So the nervous system is able to connect to lots of different targets. We often think of them just as communicating with other neurons, but that's not the case. Could be a gland, could be a muscle, could be whatever. You'd have a hard time finding any examples of things that the nervous system doesn't innervate, frankly. So let's take a look at the ligands that act as neurotransmitters. They typically are hydrophilic, so they need a cell surface receptor in order to transduce. They're not like a steroid hormone that's hydrophobic and can slip across the membrane, or a gas for that matter that could do that. So these need a surface receptor on the receiving cell or the target cell. So the way these work, they're packaged into these synaptic vesicles. Those vesicles fuse with the plasma membrane, and now we release their contents, the neurotransmitter, into the synapse. And the transmitter will come across the synapse, bind to its receptor, and then cause a shape change in the receptor. This allows the receptor to open, and now ions flow into the receiving cell. Often, this is what we call ionotropic signaling. Ion moving is what that means. And that's just to remind you that it's actually ions that move through those channels, not the neurotransmitter itself. So what kinds of hydrophilic molecules can do neurotransmission? It's, amino acids, simple ones like glutamate, for example, can be neurotransmitters. I bet you didn't know that amino acids can also be neurotransmitters. That's kind of bizarre. Or, things like acetylcholine, dopamine, serotonin, etc. What they all have in common is they're hydrophilic. And they're all released when an action potential invades the axon of the presynaptic cell. Now, later on, we'll see there's two different types of receptors that neurotransmitters can bind to. I've focused on the ionotropic variety here. Later on, we'll focus on something called the G protein coupled variety. All right, second to last slide for part 1. Here's another type of ligand. I told you it was a laundry list. So now we're dealing with what we're calling the peptide hormones. So steroid hormones, we talked about first. Those are the hydrophobic ones. Peptide hormones are typically hydrophilic. So they're not going to be able to slip across the membrane. They're going to need a cell surface receptor. These are highly variable in terms of their size. They range anywhere from a few amino acids. I mean, look at this one. Here's a peptide hormone called enkephalin that's only 5 amino acids long. So it's a tiny little peptide. All the way up to, pretty large molecules like these growth hormones that are 190 plus amino acids. So they're quite variable in their length. Here you can see some other examples. Insulin, the 2 different types, a couple dozen, less than that, 21 and 30. So what I'm saying here is that these peptide hormones are actual peptides, and they're typically products of gene expression. So remember, you have a gene, it gets transcribed into an mRNA, that gets translated into a protein. So the peptide hormones are made by this gene expression pathway, and activation of those genes is what allows the ligand to be produced. So I'm not going to tell you too much more about these now. We're going to just briefly mention the role that they play in analgesia, which is relief from pain. If you've ever heard of the endorphins, the so-called endogenous opiates that help to tone down pain or act in an analgesic way, we're talking about beta endorphin, enkephalin, and others. They are an example of a peptide hormone. And if you've ever heard of nerve growth factor, that's another example of a peptide hormone. This is something we'll talk about later in our class. All right, last one for part 1, the last type of ligand that we're discussing today. And this is a lipid. This is another one where when I learned about this, it was quite bizarre. Couldn't quite wrap my head around it at first, but now it makes sense. So yeah, it turns out lipids can act as ligands too. And this is often autocrine. It can also sometimes be paracrine. And the best connection that you and I have to lipids as a ligand is inflammation. So that's the example that I'm going to cover with you. So we're all familiar with inflammation. We sprain our ankle and it hurts. We feel the pain from that, obviously. But then we start noticing changes in the sprained ankle, like it starts to swell up. It starts to turn really red. It's starting to stiffen up. Many of these changes are, particularly the turning red and the swelling, are because of inflammation. So the tissue turns red because the blood vessels dilate to allow, things to get in there. Things like anything that needs to clot, anything that needs to repair, components of the immune system, etc. And it starts to swell for a similar reason. If you have more stuff going in that direction, then it's going to get larger. So how do we control inflammation? Well, you put ice on it, you elevate it, and you probably take NSAIDs, the non-steroidal anti-inflammatory drugs like either aspirin or ibuprofen. What you're trying to do with these NSAIDs is you're trying to tone down the inflammation. So how does all this work? Well, it turns out that in the membrane there are particular types of phospholipids that can be acted on by enzymes. Those enzymes, like these phospholipases, are stimulated by inflammatory stimuli. So inflammatory stimuli that are invading that tissue coming in from the blood are able to activate these enzymes, and they result in the production of something called arachidonic acid. So different types of arachidonic acids, like these prostaglandins, are now in the membrane. And other enzymes, these COX or COX enzymes, will convert one type of prostaglandin into another type of prostaglandin. There's 2 types of these COX enzymes, COX-1 and COX-2. Okay? And what happens with COX-2 production of prostaglandin H2 is we get even more inflammation. So other inflammation mediaries are produced from the cell, and they can then feed forward in a positive feedback loop to cause more inflammation. So it's useful to inhibit this step. We'd use aspirin or ibuprofen to block this enzyme, and that helps to tone down the inflammation and helps to cut out the positive feedback loop and helps to shut down the sort of prime mover of this pathway. That's very useful. if you've ever taken ibuprofen or aspirin over a period of many, many days at high levels, you might have noticed you're getting a stomach ache and some GI disturbance. And the reason for that is aspirin and NSAIDs are non-selective COX inhibitors. Not only do they inhibit COX-2, which is the one that leads to inflammation, they also inhibit COX-1. Well, this one we don't necessarily want to inhibit because of its homeostatic functions, like building up the lining of the stomach and the GI tract. So if you take a lot of aspirin or NSAIDs, you're inhibiting COX-1, you're not building up rather the GI lining very well, and now it's more sensitive to acid. That gives you the stomach ache. So companies, of course, are smart about this. They say, well, hey, we'd love to be able to inhibit COX-2 and leave COX-1 alone. So drugs like Celebrex and others made by companies like Pfizer are able to selectively inhibit the one we want to inhibit, COX-2, but they leave this one alone. Okay. So anyway, inflammation and the use of arachidonic acid and prostaglandins, an example of lipids acting as ligands to induce effects in a receiving cell. All right, that's the end of part 1. I'll be back shortly to finish up with part 2.